Hi, my name is Emily Gray, and today we will be talking about the differential localization and anabolic responsiveness of mTOR complexes in human skeletal muscle in response to feeding and exercise. mTOR stands for mammalian target of rapamycin, and it's a highly conserved serine threonine kinase in all eukaryotes. Its discovery changed the way scientists thought about cell growth. It was originally thought that cell growth occurred whenever building blocks were available, but the discovery of mTOR led scientists to believe that cell growth is highly regulated. mTOR is a master growth regulator, its most well-known function involving its response to extracellular stimuli, as well as cellular energy and stress. mTOR activates protein synthetic machinery in cells to increase cell growth and metabolism. mTOR is regulated through extremely complex signaling pathways, predominantly by many phosphorylation events involving multiple phosphorylation sites. Its regulation is characterized by feedback loops and many branched pathways. mTOR functions in two multi-protein complexes, both of which we will talk about next. The two complexes that mTOR reside in are mammalian target of rapamycin 1 and 2, abbreviated as mTORC1 and mTORC2. These multi-protein complexes have both different structural components as well as functionality. However, both of them contain the mTOR serine threonine kinase. mTORC1 is considerably involved in regulating transcription and translation of protein machinery to influence cell growth, proliferation, and survival, and its function is known significantly more than mTORC2. Because it is involved in cell growth and proliferation, it is directly involved in muscle protein synthesis, notably after muscle stimulation by exercise. mTOR2 is a much less studied complex. However, we do know that it is involved in rearranging the cytoskeleton in times of cell survival, and it is also involved in insulin sensitivity. Some important information had been discovered prior to this experiment. First, it was discovered that mTORC1 must co-localize with a particular lysosome in order to function, as inferred by the phosphorylation of S6K1 on mTORC1. It was also discovered that the localization of this protein lysosome complex to the cell periphery was crucial to the activation and function of mTOR. The prevalence of mTORC1 has on cell growth and proliferation in skeletal muscle cells will be the major focus of this presentation. There are multiple factors that influence mTORC1 activity, such as the presence of amino acids, oxygen, ATP, and glucose, all building blocks necessary for cell growth. The activation of mTORC1 ultimately leads to final production of proteins, membranes, and nucleic acids. These macromolecules are all crucial to cell growth and proliferation. The study we will be focusing on today was aimed at evaluating mTOR translocation in the cell to determine if it is specific to mTORC1 or mTORC2. They also wanted to study if there was a synergistic effect of acute resistance and protein feeding on the mTOR lysosome complex, or mTOR LAMP2, co-localization and translocation to the periphery. The researchers hypothesized that the translocation was specific to mTORC1 given the previous findings on the functionality of mTORC1 in muscle growth, and also hypothesized that there would be a synergistic effect of mTOR LAMP2 co-localization with both acute exercise followed by consumption of protein and carbohydrates compared to just consumption alone. This experiment was performed by using 8 healthy active males, all of which fasted for 10 hours prior to the experiment. A biopsy was taken from the vastus lateralis of all the men prior to treatments. They were then subjected to 4 sets of unilateral leg extensions, followed by immediate consumption of Gatorade Recover, which contained 20 grams of protein, 44 grams of carbohydrate, and 1 gram of fat. Biopsies were then taken from the men 1 hour following consumption and 3 hours following consumption. An immunohistochemistry assay was set up by examining approximately 120 muscle fibers from each time point and using monoclonal antibodies specific to mTOR and LAMP2. They used wheat germ agglutinin to visualize the cell periphery. They then used imaging software to visualize the tissue pre-treatment, 1 hour after treatment, and 3 hours after treatment of either exercise and consumption or just consumption alone. These images were then quantified with Image Pro software. The experiment led to multiple conclusions regarding mTOR activity. First, the predominant complex involved in response to exercise and protein carbohydrate consumption is mTOR1. They also determined that mTOR lysosome translocation to the periphery occurs in parallel to S6K1 activity on mTOR1. Finally, as we will discuss next, resistance combined with protein carbohydrate consumption works synergistically to increase mTOR1 lysosome translocation to the cell periphery and amplifies its effects. Figure 2 from this paper focuses on the mTOR co-localization with LAMP2 as well as the translocation of mTOR to the cell periphery. Panel A is a visualization of the images after treatment with the antibodies. mTOR is seen in red, LAMP2 in green, and wheat germ agglutinin in blue. Panel B quantified the LAMP2 intensity. Intensity was measured by pixel intensity on the y-axis and treatment times on the x-axis. Intensity was taken at pre-treatment, 1 hour post-treatment, and 3 hours post-treatment for the fed and ex-fed conditions. At 1 hour in both fed and ex-fed conditions, LAMP2 intensity increased. At 3 hours, LAMP2 intensity decreased in the fed treatment to baseline levels will continue to increase in ex-fed treatment. Panel C quantifies the co-localization of mTOR with LAMP2 pre-treatment, 1 hour following treatment, and 3 hours following treatment in both fed and ex-fed conditions. After 3 hours, the co-localization of mTOR with LAMP2 was greater in fed conditions than ex-fed conditions, which was an unexpected result, as researchers hypothesized that the co-localization would be greater due to the synergy of protein carbohydrate consumption with exercise. Panel D quantifies the co-localization of mTOR with wheat germ agglutinin, indicating the translocation of mTOR to the cell periphery. After 3 hours, both fed and ex-fed conditions showed an increase in translocation, with ex-fed being greater than fed, which was expected due to the synergistic effects of exercise and protein carbohydrate consumption. One unexpected result seen in panel D is the decrease in fed back to baseline levels after 3 hours. Here, we can summarize the results of this experiment to put it into perspective. LAMP2 intensity was greater in ex-fed conditions compared to fed conditions, indicating that there are more lysosomes present where exercise is implemented, probably because the mTOR1 co-localization with LAMP2 is regulating cell growth after acute exercise, and mTOR1 is increased following exercise. The co-localization of mTOR with LAMP2 is greater in fed conditions after 3 hours compared to ex-fed conditions, which is an unexpected result. Finally, the translocation of mTOR LAMP2 to the cell periphery is increased after 3 hours in ex-fed conditions and is greater than fed conditions, indicating that there is synergy in both exercising and protein carbohydrate consumption causing this translocation. So the first discussion question I have today is, what is a possible explanation that the greater co-localization occurred in fed conditions compared to ex-fed conditions? Second, what are some important clinical implications of learning more about mTOR1?